Honors Biology Final Review. You can follow along with this review using your review packet, and you can access the review packet, this video, and notes at srhsbio.wikispaces.com. First section is about ecology. First question is about methyl mercury biomagnification. It asks you to create a food chain, labeling the food chain with the producer, primary consumer, secondary consumer, and tertiary consumer. So at the bottom of our food chain is our producers. In this particular example, these are going to be seagrasses and algae. So these are photosynthetic organisms that use sunlight to make food. The arrow goes in the direction of the energy flow. So the primary consumers eat the primary producers. And the animal that's going to represent this, or the organism that's going to represent this, is the periphytons. Periphytons are consumed by crayfish, and crayfish are consumed by our tertiary consumers, which is going to be a bass in this example. Now, based on the food chain, explain how methyl mercury can build up to toxic levels. So here we have our primary consumer, and this primary consumer already has 1 unit of food energy. And on the first day, this primary consumer is going to go around and it's going to eat 10 units of food energy. At the end of each day, we lose 90% of that food energy as heat to the environment. So we ate 10 units of food. We're going to see that 9 of them disappear. 1 remains. This remaining unit of food energy can be used by the organism to grow or to meet its other needs in the environment. On the next day, the producer has become contaminated with methyl mercury, so that was represented by our color change. So now when the primary consumer uptakes energy, whenever it eats, it's going to eat 10 units of food energy, but it's also going to eat 1 unit of methyl mercury, represented by that dark circle. Again, we're going to lose 90% of our food energy, but the methyl mercury will be left behind because it cannot be used for energy by the primary consumer. So we see again that 9 of those food units disappear. Okay, next day, again, we uptake energy, 10 food units and 1 methyl mercury unit. And again, we lose 90% of the food units only. 1 more day of feeding, we eat 10 units of energy plus 1 unit of methyl mercury. We lose 90% of our food energy. So at the end of this 4th day of feeding, my primary consumer has 5 units of food energy and 3 units of methyl mercury. So as it feeds, it consumes both food and methyl mercury. Each day, the primary consumer loses 90% of the energy from the food. Leftover energy is used to help this organism grow and live. Food is lost each day, but the methyl mercury is not used for energy, so it doesn't get digested. Instead, it remains behind. It gets deposited into the tissues of the primary consumer. As we move up trophic levels, we see that this process is magnified. So here is my day 4 consumer. It has 5 units of food and 3 units of mercury. Now, my secondary consumer is going to eat 4 primary consumers. So that's why we've got 4 primary consumers here, because we're going to eat 4 of them for my secondary consumer. So if we eat 4 primary consumers, and each one has 3 mercury units, we end up with 12 mercury units. Same thing for the food. We've got 5 food units per primary consumer, and there's 4 of them that are being consumed, so that's 20. So that means that when the secondary consumer eats the primary consumer, each day it's getting 12 mercury units and 20 food units. So, again, we lose 90% of our food unit energy. So this has no effect on the mercury units because those aren't digested or used for energy. Instead, they are deposited into the tissues. So if we lose 90% of 20, we end up with a loss of 18 food units. So each day, after losing the food units as heat, the secondary consumer has uptaken 12 mercury units and only 2 food units. If we continue this process for 4 days, so we're going to eat the primary consumer for 4 days, we end up with 12 times 4, which is 48 mercury units, and 2 times 4, which is 8 food units, and we started with 10, so that's where we got that 18 from. The secondary consumer had 10 to begin with and gained 8 over the 4 days of feeding. So at the end of the 4th day, my secondary consumer has 18 food units and 48 mercury units. We're going to repeat this process, except this time my secondary consumer is going to be eaten by a tertiary consumer. So one of the day 4 secondary consumers has 48 mercury units and 18 food units. My tertiary consumer is going to eat 2 secondary consumers per day. So that means he would uptake 96 of the mercury units, 2 times 48, and 36 food units, 2 times 18. We're multiplying it by 2, again, because we're eating 2 secondary consumers per day. So that means that each day, the tertiary consumer is going to consume 96 mercury units and 36 food units. Once again, we lose 90% of our food units as heat to the environment. There is no effect on the mercury units. If we lose 90% of 36, that's going to be roughly 32, which means that each day that the tertiary consumer eats the secondary consumers, it obtains 96 mercury units and 4 food units. If we repeat this for a total of 4 days of feeding, we end up with a tertiary consumer that has 384 mercury units and 36 food units. And again, 4 times 4 for 4 days gives me 16, and we started with 20, so that's where we get that 36 from. For 96, we just multiplied that by 4 to get 384. So my tertiary consumer has 36 food units and 384 mercury units at the end of day 4. So as we move up these tropic levels from primary, secondary to tertiary consumer, we see that the amount of methyl mercury starts to exponentially increase. So a dietary recommendation for at-risk groups. So first would be to eat at lower tropic levels to get the benefits of seafood. So for our primary consumer, that has the least amount of mercury units compared to the food units. So that would be better to consume a primary or secondary consumer as opposed to this tertiary consumer that's got tons and tons of mercury in it. Avoid eating these tertiary consumers. So these are going to be like tuna, game fish, the large predatory fish that are in the ecosystem. Avoid eating those to avoid eating large amounts of methyl mercury. Another option might be to eat younger fish. Younger fish have been feeding in the environment for less time, therefore they have not been able to accumulate as much methyl mercury. Next question is about a food web. So the question says, what would happen in the ecosystem if the amount of carbon dioxide in the atmosphere was to decrease? So, of course, carbon dioxide, that is what we need for photosynthesis. So if there is less carbon dioxide available, that means less photosynthesis. Less photosynthesis means that there's going to be less biomass at the producer level. So there will be less plants. The plants are not going to be as productive. So there's not going to be as much food for the successive higher levels in the ecosystem. So with less producers, that means that fewer consumers will be supported by the environment. So the next question asks, what would happen to the prairie ecosystem if the amount of sunlight was to decrease? Again, sunlight is needed for photosynthesis. So with less sunlight, we have less photosynthesis. So it means that we'll have fewer plants. There'll be less biomass at the bottom of the pyramid. So fewer producers means fewer consumers. So if I make the base of my pyramid smaller, all the layers on top of that base are going to have to be made smaller too. So photosynthesis, plants require carbon, water, and sunlight to make sugars and oxygen. So here's the formula for that. I have carbon dioxide with water and light, and we're going to produce sugars. So this is glucose, it's a sugar, and we also produce oxygen. So my question for you is, based on the last slide, which of the following species is likely a photosynthetic producer? So in this graph, we have along the y-axis my population size, and then along the x-axis I have the water depth. And you can see that there is a relationship here between the size of the population and how deep we are in the water. So if we look at something like species C, which is right here, species C is pretty much constant. So species C doesn't have much relationship between population size and water depth. If we look at the species D here, we see that we've got the biggest population at a medium water depth, and then it kind of drops off more or less in each direction. So what is it about water depth that relates to photosynthesis? Well, the deeper down we are in the water, the less sunlight can penetrate. So any photosynthetic producers are going to get less sunlight the deeper down they are. So we would expect that for something that is photosynthetic, as we go down in the water column, the population should get smaller too, because there's less sunlight available for photosynthesis. So that is going to be species B. That must be photosynthetic, because we see the correct relationship there between water depth and the population size. Next question asks about what would happen in our prairie ecosystem if an invasive species of locusts were to appear. So we see in our ecosystem we do have some locusts that are part of this native population. But what this question is asking is if some other species of locust that wasn't native to this ecosystem were to suddenly be introduced. So this would decrease biodiversity. What would happen whenever that locust move into the ecosystem? It would take over the niche of the native locusts. And it might not have any predators, and this could lead to some uncontrolled population growth, which means that these locusts, which feed on plants, would be eating up all of the producers in the ecosystem. And again, if that bottom level gets smaller, the levels on top in the pyramid must also get smaller. So it might wipe out plants by eating them, and this could lead to other species dying off because they don't have access to those producers any longer. So this would decrease biodiversity. We would have fewer species than what we started with, and we would see that those producers would just get wiped out by this uncontrolled locust population that would be an invasive species. Next question is about the differences between primary and secondary succession. So succession is what happens whenever an ecosystem is progressing towards that climax community. So that climax community is going to be the community with the most species, the greatest biodiversity. So primary succession is where we begin with bare rock and we develop into that climax community. There's no soil present whenever we start, and this takes thousands of years. So if you look at this picture, we start with bare rock, and then we have the lichens and mosses. These guys attach to the rock, they grow on the rock, and they break it down and start to make soil. Once that soil starts to appear, we get our grasses, and again, those grasses are preparing the ecosystem for the next step where we get small shrubs, we might start to get fungi, and over time, eventually we develop this climax community of this big forest with lots of different species and lots of different diversity. Notice that as species move in, they make conditions that prepare the environment for the next species to move in. So these mosses and lichens, these are called pioneer species because they're the first species to move into the ecosystem and prepare it for the successive generations of plants and animals. Secondary succession is a little quicker because we begin after the previous ecosystem is destroyed. So maybe there is a fire or a flood, and we still have soil that's left behind, and there might even be some plants that are left behind or seeds that could quickly develop and grow into plants. This takes hundreds of years as opposed to thousands of years. So here we have an ecosystem that's been wiped out after a fire, and there's still some plants left behind. There's some soil there. Those plants fill in and grow, and over a shorter period of time, we again end up with this climax community that is filled with many different species with lots of biodiversity. Next question is about the relationships between organisms in an ecosystem. First, we have predation. So predation is where one organism kills and eats another organism for food. And an example of this would be like wolves hunting and capturing and killing and eating an elk. Mutualism, so a mutual relationship is where both organisms benefit. An example of this might be bees and flowers. So as a bee goes from flower to flower, it's collecting nectar, and it takes that back to its beehive, which it uses to make honey. But while the bee is foraging, it's also spreading around pollen from the flowers. So this allows the flowers to reproduce, it helps spread their pollen around, and both organisms are benefiting. In commensalism, this is where one organism benefits while the other one is neither helped nor harmed. So an example would be barnacles living on a whale. Because the whale is moving and the barnacles are attached to it, there's lots of water going by the barnacles, and that water is filled with food particles. So by growing on this whale, these barnacles are able to capture much more food. The barnacles don't do anything to the whale. They don't hurt it or help it. It might look like they might be harming it because they're growing all over the whale, but there's no harm done to the whale. The whale doesn't affect its ability to get food or mates or any of the other things that whales need to do. Parasitism is where one organism benefits, and that's going to be the parasite, while the other is weakened, and that will be the host. So an example of that could be like leeches or ticks attaching to the dog. Those parasites feed off the blood of the dog. This takes away nutrients that should be available to the dog, but instead are being soaked up by the parasites. And they also usually transmit diseases and things of that nature. So definitely a relationship where the dog or the host is being harmed or weakened by the parasite. So the next question asks, what essential component is missing from this ecosystem? So down here at the bottom, we've got our trees and grasses. Those are my producers. And then I've got my deer, rabbit, mouse, and crickets. Those guys are going to be consumers. The mouse, since it also eats the cricket, would be a secondary consumer. Same thing. The frog is also a secondary consumer. The snake and the mountain lion, according to this food web, those are secondary consumers, and the hawk would be a tertiary consumer because it eats the frog, a secondary consumer. So what we're missing in this organism, or in this food web, is an organism which can take all of these nutrients in these animals once they're dead and recycle them so that they can be reused by the producers. So we are missing decomposers. So if we add that on, we would draw a line from each animal in this ecosystem to the decomposers, because once these things die, it is the job of the decomposers to break them down. So this helps cycling of nutrients and materials between organisms and their environment. Otherwise, once these organisms died, all of those nutrients would be locked away and they would be inaccessible to other things in the environment. So an example could be bacteria, which helps to restore nitrogen compounds to the soil. So what role do bacteria play in the nitrogen cycle? So nitrogen is essential for life. It's in amino acids, it's in proteins, it's in DNA, genetic material, so it is an important element. Nitrogen is available in the air. The air is 70% roughly nitrogen, but it must be fixed into another form so that it can be used by living organisms. So nitrogen, as it exists in the atmosphere, is not something that organisms can use and access. So this process is called nitrogen fixation, and about 90% of it is done by bacteria in the soil. So this is essential because without these bacteria, none of that nitrogen that's needed for amino acids and proteins, none of that would be available to any of these organisms. So the bacteria fix the nitrogen, And usually this is taken up by plants, which is then taken up by our primary consumers all the way up the food chain. Scavengers, why are they important? So scavengers feed on dead animals. So basically what this does is it kind of speeds up and aids the recycling of environmental materials and nutrients. So this is the job of bacteria and detrivore decomposers, but the scavengers begin this process, and the decomposers and detrivores then complete it once the scavengers have started it. So it kind of speeds things up. Those scavengers break down that dead, decaying matter, and then the decomposers and detrivores can move in. So common scavengers, we think of vultures and hyenas. So those are things that typically are going to feed upon things that are already dead. They're opportunists, so they're looking for any opportunity to scavenge off of something that is already dead. So next question is about food pyramids and biomass. So again, biomass is just the mass of all of the living things at a particular trophic level. So at the bottom, the base has the greatest biomass, and those are going to be our plants or our consumers. In this particular ecosystem, the plants would then be consumed by small fish, which would be consumed by large fish, which then could be consumed by birds. So each level can only keep 10% of the energy that it gets. The other 90% is lost as heat to the environment. So because less energy is available at each level, less biomass can be supported at each level. So to support the upper levels with enough energy, we must have the bottom of the pyramid as the largest. Section 4 is about evolution. So number 18 asks us to compare natural and artificial selection. Describe what roles humans play in artificial selection and compare these to natural processes that take place during natural selection. So in natural selection, the environment has pressure on which organisms are going to be best suited. So the organisms that are best suited to their environment, those are the ones that are going to survive and reproduce. So perhaps these particular organisms are better at blending in, perhaps they can get more food, perhaps they are better at attracting mates. But one way or the other, they're going to survive longer, which means they will reproduce more. So their genes are going to be passed on to more offspring, so that gene will become more common in the population. The fastest natural selection is going to occur whenever there's sexual reproduction, so that means lots of genetic variation in a changing environment. So the changing environment means that the pressure acting upon that population is going to be changing. So as the pressure changes, only those organisms that are best suited to the environment will be able to survive. Next question is about comparing these. So when humans artificially select traits, they are acting as the environmental pressure. They select the desirable traits, and organisms that have undesirable traits are not allowed to reproduce. So this is how many varieties of dogs and crops were artificially selected by humans. So in our little picture here, whenever humans select the traits, it may not be the traits that are best for the environment, it just might be traits that we like. So if we're looking for a small, cute little dog, obviously that probably would not be best suited for nature. Next question asks us about a common ancestry. So first is embryological studies. So if we look at the embryos of different organisms, we see that there are similar patterns of development. So we'll see that eyes develop at around the same time, or limbs develop at around the same time. So this implies that there is some kind of relation and that they have common ancestors, because the basic plan of an organism's beginning remains the same for many different species. So here we have a fish, a reptile, a bird, and a human, and they all have gill slits. Now that doesn't mean that humans have gills, or that at one point we had gills whenever we were in the womb. It just means that in fish, these gill slits eventually develop into gills, whereas in humans, that structure develops into something different. But again, very similar early on in embryological development. So the next question is about DNA similarities. So the more DNA organisms have in common, the more closely related they are. So whenever scientists first went about categorizing different organisms, it was based on what those organisms looked like, or perhaps how they lived in their environment. Now that we have DNA technology, we're able to actually look at how closely related genetically different species are. So this is the strongest evidence for evolution. So for example, human and chimpanzees share about 98% of the same DNA sequence. So in this picture right here, I have the percent DNA in common for several species. So we see that species A and B have very little DNA in common, so they're probably not very closely related. Species A and E, however, have a high percentage of DNA in common. So A is more closely related to E than it is to any of the other species. Next question is about homologous structures. So these are going to be structures that have the same structure, but they have different functions. So the common ancestry of species can be seen in the structure and the development of these homologous structures, even if their function is different. So if we look at these different organisms here, we have a penguin, alligator, bat, human, cat, whale. So we see that the more closely related the animals are, the more similar their structures. So if you look at the mammals, which are along the bottom, the human, the cat, the whale, and the bat, we see that those structures are much more similar than they are, say, for the penguin. Next one is about amino acid sequences. So DNA codes for proteins. The DNA gives us the amino acid sequence. So because just the way that DNA shows how closely related two organisms are, so does amino acid sequences. So the more closely related two species are, the more similar their proteins, their amino acid sequences, are going to be. Next question is about forming new species. So there's a population of organisms that are permanently split by a natural barrier. The two environments are going to be significantly different. So explain how this could result in two different species. So in my picture here, I've got a population of pine trees, and they become separated by this river that suddenly forms. So on the two different sides of this river, the two species are experiencing different environmental or selection processes. So over time, traits will change to the ones that best match the separate habitats. So perhaps there's something different about the soil quality here, or the amount of sunlight, or something like that. So these two species of pine trees begin to develop. And it's again because they're experiencing those different environmental or selection pressures. Over a very long period of time, these two separate populations of trees may have changed so much they are no longer able to mate with each other. So at that point, they're going to be two separate species. So what is a species? A group of organisms that are similar and they are capable of interbreeding. So obviously if we try to mix a fish with a gorilla, that's not going to work. So only members of the same species are able to interbreed with each other. Next question asks, how do genes, traits, and evolution all connect? So evolution occurs through natural selection. So certain individuals will be selected based on the traits that are best suited to the environment. The individuals with these best suited traits are going to live longer. Maybe they can access food easier, or just otherwise are able to survive better. So traits are due to genetic variations, and if they are favorable, then those particular organisms will be able to survive. So an example would be gray fur, brown fur, white fur. Fur is present on all of the rabbits, but there is variation in the color of the fur, and perhaps those white rabbits are better able to survive the winter because predators can't see them against the light snow. So maybe the white rabbit fur would be a more common trait in the population. This means that these individuals are able to reproduce more. So in other words, they're able to pass on those best suited traits through their genes. So over time, the population will be affected as a whole. So the frequency of certain traits will change depending on which traits best match the environment. So this will lead to evolutionary change. How can exposure to an antibiotic lead to resistant bacteria? So here I have a colony of bacteria, and they're all the same species, but there's some slight variation between them, and that's represented by the different colors. So this colony of bacteria show genetic variation. Now I'm going to treat the bacteria with an antibiotic, and we see that most of the bacteria die off, but a couple of them happen to have some slight resistance due to that genetic variation. Now, that bacteria, the surviving bacteria, repopulates, and now the colony is resistant to the antibiotic. So if I apply the antibiotic, no effect. Now this doesn't just happen in one generation, it's usually due to many generations of bacteria being treated by an antibiotic. So that's why if you have a cold, the antibiotic is not going to do anything, one, because a cold is caused by a virus, but you could also be making resistant bacteria. So that's generally not good. So we try to avoid prescribing antibiotics unless a patient really needs them. And because these bacteria produce asexually, once that resistance is acquired, it is usually kept through successive generations of bacteria. 24 is to define and give an example of an adaptation. So an adaptation is just some trait which helps to increase an organism's fitness to the environment. So it is developed and maintained by natural selection. So an example could be thick fur and a layer of fat to help an organism survive in cold climates, such as this polar bear who really wants you to stop messing with his ice. Last section is called skills and processes. So give an example of a bias in an experiment. So bias is a really big problem in experiments. Scientists are always trying to remove bias to make their experiment as objective as possible. So bias is whenever you lean towards something or you do something that alters the outcome of an experiment to meet your assumptions or to meet your needs. So an example would be telling the patient in a drug study that he should expect hair growth. So he might start seeing hair growth even though there really isn't any going on, because now he expects that. That's his assumption from the experiment. Another example could be only publishing the results that meet your needs. So a drug company might hide all of the studies that show negative side effects from a drug and might only publish the ones that show the drug working. A placebo or sugar pill can help to eliminate bias. So if it's a drug study, you give the patient a placebo. That way, anything that those patients report, you're able to compare that to the things that the patients who did take the actual drug reported, and you can eliminate bias through that means. Next question is about the size of a cell. So a child is 1 meter tall, making him 10,000 times taller than a typical animal cell. So what is the size of an animal cell in meters? So basically, a child is 10,000 times bigger than an animal cell. So if we count the zeros, we've got 4 zeros. So if I were to write that in scientific notation, it would be 1 times 10 to the negative 4. The reason it is negative is because the cell is smaller than the child. So if I asked how many times bigger is the child than the cell, it would be 10 to the positive 4. But because the cell is smaller, we get negative 4.